So this is what a standard data set in an fMRI experiment might look like. Typically, you would have one or more structural images. So this would perhaps be a T1 weighted image, just to give you a detailed view of the anatomy of the brain of your participant. And then you might have one or more functional images. In our case, these will be T2 star weighted images. And in this particular case, I'm just portraying, let's say, 4. And each of these images, as you know well, is typically acquired in a number of slices. And also worth pointing out, whereas the structural image is typically highly detailed and has high resolution, the functional image usually has lower resolution. When thinking about functional data, there are typically two different ways of conceiving of these data, or there are two different intuitions in terms of representing these data. One is more spatial in nature, and one is more temporal in nature. But throughout this lecture and the following ones, it'll be convenient to sometimes think about one of the two, and sometimes think about it with the other representation. So I want you to have both intuitions, and I'm going to portray on this slide both intuitions. Now, one intuition is more spatial in nature and views MRI data and fMRI data as a more spatial thing. So typically, when we acquire one data set, what we're doing is we're acquiring a series of images of the brain, each one of which we refer to as a volume. So if I say a volume of data, I literally mean one image of the brain. And as you know well, each image of the brain is made of voxels, small cubes. And inside each of those small cubes, there is the value of how strong M0 is when you snap the picture, so at TE. You remember, protons, radio frequency pulse, they go in the transversal plane, they start dephasing, and then you capture a picture of how strong M0 is in the transversal plane at the echo. That's the number that goes inside each one of the cubes inside a volume. And then, as I was saying, we acquire one such image every so often, which is every TR, or at every time of repetition. For example, every 2 seconds. Now, the set of data, so all of these images from when we start the MRI machine to when we stop it. So from when we start the data acquisition, and typically our participant engages in some task, and then we say to the participant, okay, thank you, you're done. We stop the data acquisition. We refer to that as a run. Okay, so a volume is a single image. A run is usually a consecutive collection of data points. So one intuition, this more spatial intuition, essentially boils down to thinking of functional data as a set of volumes taken one after the other. And inside each of the cubes of each of this volume, there are numbers of how strong M0 was at the TE. A different way of representing the exact same information, so these are the same data, I'm just showing them in different ways, is to imagine that what we're doing is collecting a volume of data every 2 seconds. And so you could imagine that essentially each of these voxels has attached to it a time series. So this up here is the value of this particular voxel at the first volume. And then this is the value of that same voxel at the second volume. And then this is the value of that same voxel at the third volume, and so on and so forth. And the second row is the values of a different voxel at each of the TRs, et cetera, et cetera. So sometimes it will be handy to think about a functional data set or a run of data as essentially a set of pictures of the brain over time. Other times, it will be more handy to think about these data as time series, where each voxel has a time series of values over time. They're the exact same thing, I'm just portraying it in different ways. Now, here's a typical sequence. Let's say you perform an experiment. This is the typical sequence of in which you would analyze your data. So you might acquire the data, again, in our case, 1 structural image and 4 functional images. Then what you would do is you would do pre-processing to the functional data. And that's what we are going to focus on today. Once you've pre-processed the data, you will do the analysis at the single run and single subject level. So you take the data for one run and you analyze that, and you see if some part of the brain has the signal that becomes stronger and weaker together with some mental activity of interest. Then we take the data from each participant, we put them all, you'll remember, in the common box or in a standard space, and then we do a group analysis. So we take the results from, let's say, 20 individuals, and we put them all together and do a group analysis. And what you're seeing here is a typical portrayal of a group analysis. So the areas that you're seeing here in colors are parts of the brain that are systematically active whenever the participants as a group engaged in some mental activity that was being tested. So let's start with preprocessing. So what is it and why do we do it? Well, really, if you can think of it this way, those time series that you saw, a series of values for each voxel, you can think of them as made of two main parts. One is your signal of interest, and the other one is noise. What do I mean? Those time series that you saw will go up and down together with some mental activity. That is our signal of interest. On the other hand, on top of that, there are a lot of sources of noise, which we'll discuss shortly, that kind of superimpose on that. So the aim of preprocessing is a series of data transformations, also sometimes called data conditioning, aimed at reducing these sources of noise. Essentially, we're trying to reduce as much as possible this side of it so that we can make the signal of interest as clear as possible. Now, it's important to point out that if you're doing a task-based analysis versus a task-free analysis, the sequence of this preprocessing might be different. In fact, task-based analysis overall, the series of preprocessing steps and the orders in which we do them tends to be overall fairly standardized. Still not a trivial problem in the sense whether the orders that we use are the best ones is still debated and they could still be optimized. But overall, software do implement a fairly standardized pipeline, although different software might implement slightly different pipelines. Now, in the context of task-free analysis instead, which step to apply and the order is actually much less clear and much more debated. And this is why it's just so much more important that you understand in depth what each of these preprocessing steps is doing and how it's changing your data.